The origin of life is a profound mystery, one that has fascinated scientists, philosophers, and thinkers for centuries. How did the first spark of life ignite in a world that was once nothing but a swirling mix of chemicals and energy? This question sits at the heart of one of humanity's greatest scientific quests. Evolutionary theory offers a powerful explanation for how life diversified and adapted over eons, giving rise to the incredible variety of organisms we see today. It describes the gradual changes that shaped simple life into the complex web of species inhabiting our planet. But before evolution could begin its work, life itself had to emerge from non-living matter. Can science explain how this first step happened, or does it remain an unsolved puzzle? Can unguided processes, driven solely by chance and the laws of nature, truly account for the astonishing complexity we observe in even the simplest living cell? Each cell is a miniature world, filled with intricate machinery and precise instructions. Many scientists assert that, given enough time, even the most improbable events can occur. The vastness of geological time, they argue, provides a canvas for countless possibilities to unfold. According to this view, random interactions among molecules in Earth's early environment eventually led to the formation of self-replicating structures, molecules capable of copying themselves. From these humble beginnings, all life on Earth is thought to have evolved. But is this a reasonable assumption, or does it stretch the limits of what chance alone can achieve? The debate continues, with some researchers expressing skepticism about whether randomness is enough. Can random chance truly assemble the intricate machinery of life, with its countless interdependent parts working in harmony? Or does the complexity we see suggest that something more than chance is at play? To answer this, we must delve into the realm of probabilities, examining the likelihood that the essential building blocks of life could form by pure chance, without any guiding hand or direction. It's important to distinguish between simple self-assembly, like the formation of crystals, and the highly specific, information-rich arrangements required for living systems. Crystals may form naturally, but life demands a far greater level of organization. Only by exploring these questions and weighing the evidence can we begin to assess the true plausibility of life arising without guidance, and perhaps move closer to understanding our own mysterious origins. Let's start with a fundamental building block of life: proteins. These remarkable molecules are found in every living cell, performing countless essential tasks that keep organisms alive and functioning. From building tissues and muscles to catalyzing chemical reactions, proteins are at the heart of biology. Proteins are long chains made up of smaller units called amino acids, which are linked together in a specific order. Once assembled, these chains fold into precise three-dimensional shapes, creating the unique structures that allow each protein to do its job. These intricate shapes are absolutely crucial for their function. If a protein folds incorrectly, it often can't work at all, and sometimes it can even cause disease. The exact sequence of amino acids in a protein determines how it folds, what shape it takes, and ultimately what role it plays in the cell. Even a single change in this sequence can have dramatic effects. To put this in perspective, consider a relatively simple protein composed of just 100 amino acids. While this may sound small, the complexity is staggering. There are 20 different types of amino acids available to build proteins, each with unique properties and shapes. The probability of getting the correct sequence by chance is like winning the lottery not just once, but over and over again, hundreds of times in a row. The odds are mind-bogglingly small, but the challenge doesn't stop there. The process is even more complicated than most people realize. Amino acids themselves come in two forms: left-handed and right-handed. Much like how your left and right hands are mirror images, but not identical. Yet, living organisms use only left-handed amino acids to build their proteins. This uniformity is critical for life, but it's not something that would be expected by random chance. The odds of randomly assembling a functional protein of even modest length, with the correct sequence and the right-handedness, are astronomically small. So small, in fact, that it defies imagination. All of this suggests that chance alone is an inadequate explanation for the origin of proteins, let alone the breathtaking complexity of an entire living cell. The mystery of how life began remains one of science's greatest challenges. Proteins are more than just complex molecules. They act as coded instructions. The specific sequence of amino acids carries information, much like the letters in a sentence convey meaning. This information determines the protein's shape and function, allowing it to perform its role within the cell. This concept of biological information is crucial. It distinguishes living systems from non-living matter. A rock might have a complex mineral composition, but it doesn't contain coded instructions. A protein, on the other hand, is a product of information encoded in DNA. This raises a critical question. Where does this information come from? Information is fundamentally different from matter and energy. It requires a source and intelligence to generate and arrange it in a meaningful way. In our everyday experience, we find that codes and information always originate from intelligent minds. Software comes from programmers. Blueprints come from architects. Encrypted messages come from code makers. We don't find complex codes arising spontaneously from natural processes. Yet, the DNA molecule within our cells contains a code far more complex than any human-made system. This code dictates the production of proteins, the building blocks and workhorses of the cell. It governs the intricate processes of life itself. Is it reasonable to suggest that the information in DNA, which dwarfs any human achievement, arose merely by chance? Or does the principle of cause and effect suggest a different explanation? The remarkable complexity of biological information points strongly towards an intelligent source. The challenges to unguided evolution extend beyond individual molecules like proteins. Life, depends on these intricate systems with numerous interacting parts. These systems are what we call irreducibly complex. This basically means that if you remove even one part, the entire system just ceases to function. Consider the bacterial flagellum, for example, a whip-like tail that propels bacteria. It actually functions like a rotary motor, complete with a drive shaft, rotor, and a flexible hook. All these parts are essential for its operation. Without any one of them, the flagellum would honestly be useless for locomotion. So, how could such a system evolve gradually through random mutations and natural selection? Each component by itself, it offers no advantage. It's only when all the pieces are assembled in the correct order that the system actually functions. This presents a major obstacle for unguided evolution to overcome. Proponents of unguided evolution often invoke natural selection as the driving force behind life's complexity. However, natural selection has limits. It can only act on existing variation within a population. It cannot create new features or systems from scratch. Moreover, natural selection requires replication and inheritance. It works by favoring organisms that are better adapted to their environment, allowing them to survive longer and reproduce more successfully. But before life exists, there is no replication, no inheritance, and thus no engine for natural selection to act upon. This presents a significant chicken and egg problem for unguided evolution. Natural selection, the very mechanism proposed to build complexity, cannot operate until there is already a self-replicating system in place. This, honestly, casts doubt on its ability to explain the origin of life itself. Mutations, random changes in DNA sequence, are often touted as the source of new genetic information. However, mutations are typically harmful or neutral. While some beneficial mutations occur, they usually involve minor adjustments to existing systems, not the creation of entirely new, complex features. Imagine a car factory. Mutations might change the car's color, slightly alter the engine's efficiency, or modify the shape of the seats. But, no amount of random tinkering is likely to produce a fully functional airplane from a car factory. Similarly, mutations might explain variations within a species, or even the development of new species from existing ones. However, they fall short of explaining the origin of fundamentally new, complex structures and information-rich systems that are the hallmarks of life. When we examine the fossil record and the incredible diversity of life on Earth, we observe clear and compelling evidence of adaptation and variation within existing kinds of organisms. Over time, species display remarkable flexibility, adjusting to their environments in ways that help them survive and thrive. These changes are often subtle, involving shifts in color, size, or behavior, and they demonstrate the capacity of life to respond to changing conditions. This process is often referred to as microevolution. Microevolution describes the small-scale changes that occur within a species or population, such as the development of new traits or the loss of old ones, usually in response to environmental pressures or genetic variation. However, when we look for evidence of large-scale transformations, those that would be required to produce fundamentally new biological systems, body plans, or entirely new types of organisms, we encounter a striking lack of evidence. This is what scientists refer to as macroevolution, and it involves changes that go far beyond the minor adjustments seen in microevolution. For example, we see finches' beaks adapting to different food sources on the Galapagos Islands, a classic case of microevolution in action. The beaks change shape and size depending on the available seeds and insects, allowing the birds to exploit new ecological niches. Similarly, we observe bacteria developing resistance to antibiotics, a process that can happen rapidly as bacteria adapt to survive in the presence of new drugs. This is another clear example of adaptation within an existing system. We also see a wide range of variations in the size, shape, and appearance of dog breeds, all of which have been produced through selective breeding, a form of guided microevolution that works with existing genetic material. These are all examples of microevolution, changes that occur within existing blueprints, utilizing the genetic information already present in a population. They show how life can adapt and diversify, but they do not demonstrate the creation of entirely new systems or body plans. What we don't see in the fossil record are the numerous transitional forms that would be expected if single-celled organisms gradually evolved into complex plants and animals. The gaps between major groups remain, with few, if any, fossils showing the step-by-step development of new organs or body structures. We don't observe the gradual emergence of entirely new organs, new body plans, or new information processing systems in the fossil record. The complexity of structures like eyes, wings, or nervous systems appears abruptly, without clear evidence of the many intermediate forms that would be expected if these features evolved through countless small steps. The fossil record, while certainly incomplete, should bear witness to such dramatic transformations if they were the result of unguided processes operating over millions of years. Yet, the expected abundance of transitional fossils is largely missing, raising important questions about the sufficiency of current evolutionary explanations. The absence of such evidence is a significant challenge to the idea that random mutations and natural selection alone can fully account for the origin of life's complexity. While adaptation and variation are well documented, the leap to entirely new systems remains one of the great mysteries in the story of life on Earth. When we step back and consider the immense improbability of life arising by chance, the exquisite complexity of biological systems, and the lack of compelling evidence for macroevolutionary transitions, a different explanation emerges as more plausible. Just as we infer the work of a designer when we see a watch or a smartphone, the intricate design of living organisms strongly suggests the work of an intelligent agent. This isn't a matter of inserting a God of the gaps explanation where science falls short. It's a matter of following the evidence where it leads. The information content of DNA, the irreducible complexity of molecular machines, and the challenges of generating genuinely new biological information all point towards an intelligence far exceeding our own. While science may never definitively prove the existence of this intelligence, the evidence from nature points more convincingly towards design than towards chance as the best explanation for the origin of life.